And there are three different things that we'll be doing this morning. The first is to finish up these slides. The second is going to be genetic transfer info. And then the third, I'll use this blank page to talk about the exam on Tuesday. So kind of do a exam topic overview. Lab today, there's about 35 minutes of video for pre-lab, and it's going to be a real similar type of lab as the last couple weeks, where you look at things from last week, do gram stain, start things for next week. So that's what to expect for today. It's another good fun lab. So when we had stopped last time, I had been trying to describe kind of how two or three of the main vaccines that the U.S. has available right now work. And I want to make a note on this page that the Pfizer vaccine and Moderna are mRNA. And I think we said that last time, that this is a good review of how that works. So there's a lipid-based vesicle, basically, that contains within it the messenger RNA. That is put into your arm in a shot. The vesicle will fuse with the plasma membrane in your muscle cells, and your ribosomes will then translate the spike proteins. And they show the spike proteins on the surface of the cell. And then you have some specialized immune cells that come and look at those and then enable an immune response, so that whatever spike protein you see there, your body detects it as foreign and then develops immune response. So with the immune response, you will eventually get to the point to have antibodies. Remember that this is going to degrade pretty quickly because it's RNA. That will not stay around long in your body, and you're just briefly exposing a person to a spike protein so that they can learn to make antibodies against it. This kind of approach, if the spike protein changes shape drastically, the antibodies that we have against it may not work. So that's the importance of the variants. If they're changing the shape enough to make this unrecognizable, our immune response may not be effective against that new shape. Need to go backwards a little bit. So this approach is the Johnson & Johnson vaccine, which just got approved last week. This is the one where there is an adenovirus vector. That word is too small on here. So the adenovirus vector is, in some aspects, a dummy, not harmful, safe virus, and it's used as a carrier to produce spike protein on its surface. So that would be the SARS-CoV-2 spike. And this dummy virus with the SARS spike is what goes into your body. This one is a little bit more robust in you don't need a booster. This is the one shot. So if you end up later this summer getting Johnson & Johnson's shot, it's just one. You don't have to go back and get another one. So I don't know, for college students that may end up being the best, because you could get it in your home state and then not worry, oh, how do I go back and get another one? That's the modified vector style. So I want to match that to what, or viral vector. viral vector is this style, and it's the Johnson & Johnson version of vaccine. This next thing that I wanted to look at is a chart of worldwide approved vaccines. It does not include Johnson & Johnson, but I want to add that on there. This was created in the UK. They already have been using a viral vector vaccine called the Oxford vaccine. That one is a 2-dose. It's 62 to 90% effective, regular fridge temp, and it's about $4. That one is pretty similar to Johnson & Johnson. That one's viral vector, except it's a 1-dose. And it's about 70% effective. It is regular fridge temperature storage. And it's about $10 a dose, fairly cheap. These two are the ones that most people in the US have gotten so far. 2-shot dose, super effective. They have to be kept really cold, so that's a challenge. Around here, if you're going to get this one, you have to go to Kingsport to get it, because they've got a lot of the super cold freezers that can keep it stable. These cost a lot more. Insurance and the government are paying for those in the US. So individuals are not paying this level for the vaccine. And then this one is particular to Russia. They named it Sputnik. That's kind of cute. It's also viral vector version, but and can go in a regular fridge. So this concept actually is why I wanted to not skip over these slides, to talk about what herd immunity is. So the first thing to know about this is what herd immunity is. And before I get to that, I want to explain how this graph is set up. So this is going across time, starting in 1996. That's when I graduated from high school. That's nice. 1996. And on this axis, in the yellow, it's showing what percent of people, and this is in the UK, what percent of people got their MMR vaccine on time. So they're calling that uptake. How many people said, yeah, I will get my MMR vaccine on time? And it's really how many parents took their kids to get MMR. No kid would decide to get MMR on time. Joseph kicked a nurse, 2 nurses actually, when he had to get his boosters. It was so embarrassing. He's really strong, couldn't hold him down, he was only 4. So the tan is like how many people didn't take their kids to get it. And then on this side, it was how many cases of measles appeared in the UK in that particular year. So the first M in MMR is measles. It's a triple vaccine against 3 different diseases. And you can see that when 90-some% of people were getting, of kids were getting vaccinated against measles, up through whatever level that is, say 85%, you did not see a rise in measles cases. They were pretty low in the population. But after the uptake dropped below about 85%, you see this pretty dramatic bump in measles cases. And then shortly after, a huge spike in measles cases, and it kept going up, and it's still pretty high. That seems like a minor drop in how many people got vaccinated from 90, I think that was like 92 to 84, something like that. But having 12% more of the population not get the vaccine is enough to let measles spread again. There are enough people not immune to allow the spread. And that's the opposite of what herd immunity is. This time frame forward, so from here on, herd immunity has been lost. And that allowed measles to come back. Measles for kids is way more dangerous than COVID. 100 times more dangerous than COVID for kids. So until then, in this section, herd immunity was working. And so what herd immunity is, I'm going to write that. Well, I don't know if I have a marker today. Yes, I do. Herd immunity is when you have enough of the population is immune to a disease so that it becomes difficult for the disease to spread. Herd immunity really is there are not enough hosts for easy spread. You can't be a host if you're immune to that disease. So you can have herd immunity in two different ways. You can have enough people get sick and survive and recover, and they can become immune that way, or you can have enough people vaccinated. It's much safer to have people vaccinated. Last fall at King, we were maybe one of the few colleges that just opened and like we're going to have class and we're going to distance and all of that. It turned out great. It turned out great. And it's possible that within like King's student population, we're actually approaching herd immunity because so many students had it and never had symptoms. Like we will never know that, but it's possible. That may be why we're having so few cases this spring, is because so many people already had it and it's not spreading very fast. That is different than letting herd immunity run through the whole U.S. population and letting everyone of every age in the U.S. get exposed. That would not be a good idea. Vaccination is a much easier way to get to herd immunity. But herd immunity, if you're doing it by vaccination, it requires pretty high uptake. And the question for SARS-CoV-2 is like, what is the vaccination rate that would encourage herd immunity? And we actually don't know. There's an estimate. So the estimates are somewhere in the 70% range. This is from last spring. May 2020, when people were getting polled, would you take the vaccine if you could get one today? And it's pretty small if you got slides printed out, but the green is not, the blue is would. And I thought this is interesting. The people who would most get the vaccine are postgraduate, like people like me that have had more education, and that makes sense because if you understand how the vaccines work, then it's not very scary as it would be if you don't know how they work. And then some groups have much lower proposed uptake than others. This has changed in a positive direction since then. Since then, if you did this poll again now, it wouldn't be as green, it would be more blue. And that's pretty much over all groups except hardcore anti-vaxxers. And some of that is because like the more people you know that have gotten vaccinated and you see their selfie thing and their little badge or whatever, like, well, maybe it's okay. So hopefully vaccine acceptance will keep going up. And if you all, when you all get a chance to get it, go ahead and get it. Even if you already had COVID, even though it's unlikely to be very dangerous to you, go ahead and get it for the good of everybody and your professors who are older. So let's see, I need to save this. And now we'll switch on to the genetic transfer slides. All right, so this, we're kind of jumping back to bacteria now. So we started with molecular genetics of microbes, and then kind of talked about viruses, individual viruses, and then vaccines. Now we're going back to bacteria. And when the first lecture in this unit, we talked about how bacteria are haploid and they reproduce asexually, and they don't really create new gene combinations. That's true for their normal process of cell divides, binary fission, make offspring. So the direct offspring are going to have whatever genes the parents had. They're not parents, the previous generation had. So there are ways that bacteria can transfer genes to each other. There are 4 ways DNA can exchange between bacteria and have the chance to get a new gene once in a while, something completely new. So the gene transfers may not be common. These 4 ways, it's not like this is happening daily for every bacterium, but when they do happen, they give a chance at something new. So if the gene that is transferred, if it's beneficial and useful, that gene can increase its prevalence by natural selection. If that gene gives an advantage to the bacteria, that will make them more likely to survive or more likely to produce a large population, and then that gene will spread throughout bacterial populations. So gene transfer in bacteria is a very big deal in giving them new abilities and changing their evolutionary history. There are 4 different ways. These 3 are the ones I'm definitely going to talk about today. If I get to transposition, I may go ahead and talk about that as well. Unfortunately, 3 of them start with TR, and they all end with shun. So it becomes a little bit more difficult to keep them straight. Don't mix up these 2. Like, they are almost the same word. Transformation, transduction. They are 2 very different things. So just be aware of that when you're studying this weekend and doing the test on Tuesday. This slide will become more useful at the end. It's an overview. So I want to start with transformation. This one is the most common amongst bacterial groups, and there's a really famous experiment that demonstrated it that you may have studied in AP Bio or Gen Bio or in high school or somewhere. and it's called Griffith's experiment. Griffith was studying fatal pneumonia in mice, and he was using Streptococcus pneumoniae. This is the one I can't get to grow in lab. I tried again this week, and it's done. It's not going to grow. We're not going to get to use it this semester. But he was studying Streptococcus pneumoniae, and there are two versions of it. There's an S strain that stands for smooth, and the smooth strain has a big capsule around it. It's got a very thick capsule, and that hides the bacteria from immune systems and makes it more deadly. So if you infect a mouse with live, living S strain, the population of mice is largely going to die. S strain is deadly. The other version of strep pneumoniae strains are designated R for rough. I shouldn't say they have no capsule, they have less capsule. Much less capsule, and they are survivable. They're survivable. So if you infect a mouse or a population of mice with living R cells, the mouse's immune system can recognize that and maybe make it the 2 or 3 weeks, make antibodies and live. Most of the mice will live if they're infected with R. So the reason that Griffith started doing this experiment is that he wanted to know what is it about S that makes them deadly and R is not. Because almost everything else about these strains, they act the same. They would give the same test results and they kind of grow the same rate. And what's the difference between these? So he was trying to figure out, are S inherently dangerous, or are they making like a toxin that goes outside of them and that's what's killing the mouse? So he decided to do an experiment where he's comparing these two first. So he took S cells and he boiled them, or he boiled them, and killed them, and then infected the populations of mice with the same number of heat-killed S cells as living ones. And he found that this let the mice live. So this told him there's not, this is not because of a diffusible toxin. It's not that the S cells are making something deadly and sending it outside of themselves and killing the mice, or at least if they are, it's destroyed by heat. He wasn't using that high of a level of heat. So, okay, it's maybe not a toxin. And he was still trying to figure this out. And then he did this experiment. He did this experiment. And why he did this, I've always been curious about. Like, why would you do that? How would you come up with that? He decided to mix rough cells, which are survivable, and heat-killed S cells, which are survivable. Both of those individually survivable, or okay. But the combo of those two was deadly. And that was a very surprising finding. Not only was it a deadly combo, but he could isolate from the dead mice living S cells. living S cells. So what he first thought is something about the rough is bringing the smooth back to life. That's what it looks like. Like if you mix dead smooth with live rough, the rough brings them back to life. That's weird. He named it transformation because the smooth cells were transformed back to living and deadly. And as he did more experiments with this, he found that DNA was allowing this. DNA was allowing the transformation process. And that took several more experiments. Well, I can't say that today. Experiments to show it. But what was happening was when you heat kill S cells, DNA spills out. DNA spills out. And if you're mixing live rough with dead S, DNA spills out. And the rough cells are using that spilled DNA. What's actually happening is the rough are using a capsule gene. And it's not that the smooth are coming back to life, it's that the rough are becoming smooth. They're becoming smooth. And they're becoming deadly enough to kill the mouse. The DNA is enabling this. So this is one of the key experiments to show that the heritable information is in DNA, not protein, or protein was the other big candidate at the time. And a lot of people didn't believe it because DNA was kind of new as a concept. But this was one of the foundational experiments to show that DNA is the heritable material. Griffith's experiment in transformation. So after many, many years of study, we have a lot more knowledge of, like, literally, how does the DNA get into the rough strain? That's what is coming up next. I'm going to skip over this. This is cool, but don't have time. It's another kind of transformation experiment. I'm going to go here. So what you're looking at on this is a cell that is going to pick up new DNA. So the purple stuff is the new DNA. And it's called the transforming DNA. It came out of some other cell that we're calling donor. So the cell that's going to pick up the DNA, its genome is colored in green. And it's going to be transformed with this piece of DNA. Transformation starts with DNA from the environment. So that DNA is from the environment. It can be a variety of lengths. And the source of that DNA is kind of irrelevant. It can be any kind of DNA as long as it's double-stranded. So what has to happen is that that DNA has to bind onto some surface proteins on the recipient to kind of hold it. And then one strand of the DNA is going to go into the cytoplasm. So we got one strand enters, the other strand is kind of destroyed during this process. And because DNA has complementary base pairing, you get all the information you need from the one strand. So the strand has to be integrated into the host chromosome if it's going to be stable and heritable. If it sticks around as a single strand in the cytoplasm, it's just going to get degraded and destroyed. It has to be integrated into the host chromosome, and then it will become part of the host's genes. And then, if that's useful, it will provide an advantage to the host. This is super, super important that you know this. 99.999% of the time, whatever DNA that was is not useful. There's no picking and choosing of like, oh, that looks like a useful gene. I think I'm going to pick that up. This is like if junk mail keeps arriving at your house and you faithfully keep opening it. There may be one time in your whole life where you're like, oh, I actually want this. Like, this is actually something good. The rest of the time it's trash. You didn't ask for it, it just kind of came there. Most of the time it's not useful. So this is not a purposeful process at all. But that, what is that, 0.001? It may even be lower than that. 0.001% of the time, that gene is advantageous. Advantageous. Another way I've described this is if you were a criminal and you went into my house and you started gathering papers from my house, there's a lot there for your choice. And out of all the 10,000 papers in my house, most of it is like elementary multiplication or coloring pages, or it's mostly papers sent home from school, honestly. But if you came across the one that is my social security card, bonanza, awesome, super great. Or if you happen to find, somebody's passport, then that's useful. But it's totally a needle in the haystack as to whether you're going to find a useful paper or a paper that's just pointless for you. That's how transformation works. Most of the time, it's not an advantage. But if it's an advantage, it's really an advantage. So looking back at Griffith's experiment, the rough cells did not decide to get capsule gene. If you think about what was happening here, there's all kinds of DNA coming out of these smooth cells, and the rough cell has almost all of it already. But all you need is one to pick up and use a capsule gene, and it can become S, and then it becomes deadly, and then it's going to survive in the mouse and not get killed. So everything that you get back out of the mice, is S, because it was a survival factor. It was the benefit to get that capsule gene. The rough strain did not choose a capsule gene on purpose. There just happened to be one that was lucky that got that gene and could use it. If the DNA is not integrated, into the host chromosome, it will degrade. It will degrade and it will not be usable. So what you're really getting is a single strand. And the single strand has to use this machinery called REC. It stands for recombination. REC-A is a protein that's very involved with this. So REC-A allows that single strand to be incorporated into the host genome and have the other strand filled in. And that's a complicated process. I don't need you to know the details of that for this class. But it's called REC-A recombination. And that will get you the pieces of donor DNA integrated and stable and be able to be passed on. It's not enough to just get the DNA into the cytoplasm. That happens a lot. Getting the recombination to integrate it is less common. But if that doesn't happen, it's like you lost the paper. You lost the important paper. Can't find it, can't use it. No benefit. I want to have the break right now before we go into the other two styles of recombination and then test review. A break right now. Kind of would like to be able to do a laser pointer thing on this. In fall 2019, I had a cell in that room across from the fish tank in Whitehall, and that has a screen kind of like this. And I was able to teach cell with screen like this, which is very nice. Better than that room with the columns. I actually really like using this. And I think it wasn't set up correctly until recently. Any of you have math in here? Is this where differential equations is? Any of you have it? Are you in differential? I think Professor Leandoo may be using this. So before I talk about the other two styles of genetic transfer, I'm going to make on this blank page kind of a comparison of the big themes of the three styles. So for transformation, you can have basically any length of DNA from any source. Any length of DNA from any source. And this approach is really easy to do in a lab setting. Like if you had molecular bio last winter or fall, I think you probably did this in a lab. Did you use the calcium method? Did you make them cold and then set the tubes?
Yes.
Yeah. It's really easy to do this in a lab setting. So once this was discovered, it really became used a lot in research because it can be any DNA. You can get it into bacteria, get them to integrate it, and then make them do your DNA work for you. The donor and the recipient don't need to be related. That we can put human genes into E. coli, and that's being done 100,000 times a day all over the world for different reasons. So transformation, there's no limit on, not much limit on how big of a piece. There is a limit, but it's a pretty big limit. The donor and the recipient don't need to be related. And the DNA can be linear or plasmid. It doesn't have to be a certain format. So transformation is kind of the easiest method. So I'll come back to that in a minute after we've talked about the other two styles. So our next style or method of gene transfer in microbes is called transduction. Transduction. And transduction is when a virus is moving the DNA. And since we're talking about bacterial cells, the virus has to be a bacterial virus. It's called a bacteriophage. And they make it look really cute in this cartoon, not the actual scary appearance that it has. So one of your quiz questions today is, what is the gray thing? It's a bacteriophage. And for transduction to happen, it's involving normal behavior of a virus, and this is just a review of that. So we've got the bacteriophage attaching, injecting the DNA, the viral DNA. I'm going to go ahead and list the stages as a kind of review. So you've got attachment and penetration. That's normal for the virus to do. The host will be then doing the synthesis of the viral parts. And then you get assembly, but the assembly is sloppy. So see here, you've got viral genomes in red and the host genome is in green. And during assembly, when the viruses are packing up to leave, one of them got host DNA in it instead of viral DNA. So this is like if I asked my little boys a few years ago to pack, like, we're going to go to grandma's, you need to pack. And at that, say like when they were 6 and 7, they would get a bag out and they would put some stuff in it, but it's like totally not the right stuff. Like it's like toys and pants, but not anything else. Yeah, I got some stuff, I'm ready to go. Viral assembly does not pay attention to what DNA goes in. It's just like, yeah, I got this much DNA, I'm ready to go. So if the host is suffering and dying and there's pieces of host DNA breaking off, they can get packed up into a virus. And one of these viruses now has host DNA by accident. This is not a purposeful process, it's a random chance kind of process. So the green virus, it's probably got all the right proteins and stuff. So now we're jumping down here. Here comes green virus, it attaches, it sends the genome in as the penetration step. This cell is really lucky. Whatever the virus just gave it, it's not going to kill it. So this cell, not only is it not going to die, it got infected by a dud virus. It's not going to die, that's supposed to be an exclamation mark. And it might get a beneficial gene or genes. It has that 1 in a million chance of getting something useful out of this. The virus is doing the movement, but what's moving is the host DNA. So this is a big driver of bacterial evolution, but it has a lot more requirements on it, a lot more requirements. So I want to go to the other blank page and put the requirements for transduction. I don't know why the pen is writing so much finer point on this, but I like it. Okay, so transduction requires a virus, a bacteriophage. That's the first part. The bacteriophage is going to have to move a fairly short length of DNA. This is limited by the size of the suitcase, so to speak. So you've got that capsid head, and it's going to pick up a certain size of DNA that's supposed to be the size of the phage genome. This is not a limitless DNA bonanza. It's going to move a fairly short length of DNA. And this cannot move DNA from E. coli to Staphylococcus because there aren't phages that infect both of those. This is limited to same type of bacterial strains. So you could move bacteria from E. coli, DNA from E. coli to E. coli, or from E. coli to E. flexneri, or whatever other kinds of Escherichia there is. It has to be within the same family of bacteria. So you got to have a virus to move it. It has to be kind of a shorter length, and the strains have to be related. it still requires integration. All of the methods require integration, but we'll write that at the end. Students really often mix up the word transduction with transformation. So maybe a dumb way to think about this is transduction. Like if the virus is sitting down on the bacteria like duck, duck, goose. Do you all even play that? Do you know what that is? I heard it had a different name in Minnesota. I don't remember what it is now. Anyway, ducking down and like squatting and injecting the DNA, maybe that's a way to remember. Transduction, transformation. The third big way is conjugation. Conjugation. So this one, at least it starts with a C. And maybe you can relate this to like conjugal visit. There's a connection, like a physical connection between the two bacteria sharing DNA. So conjugation requires a pilus. So in this method, DNA is going to move through a pilus. And this seems to be the original purpose of a pilus or pili for moving DNA between related strains. So remember, pilus is a hollow tube between two bacteria. On this picture, do not see how it says here, pilus with attached phage virions. This is not related to transduction. The only reason that the pilus has phage virions attached on it is to make it show up for the picture. That's a coincidence. There's no viral involvement in conjugation. So on this slide, just cross this out. You got these pictures with this caption, cross it out. Phages do not really have anything to do with conjugation. They're just useful for making a pilus show up on an electron micrograph. So during conjugation, there has to be a very specific kind of DNA that's moving. It has to be on a plasmid, and not just any plasmid, but a plasmid called F plasmid. And the F stands for fertility, which is a weird concept for bacteria, but that's where the F terminology came from. And the F plasmid has this certain region on it for transfer. That's what TRA stands for, transfer. And this is going to control ability to move between strains. The TRA region. And you can see that this whole plasmid is about 100 kilobases. So about 100,000 bases. If a strain has F plasmid, so having F plasmid can be denoted F plus. Like, yeah, I got it, I'm F plus. I got the F plasmid. You can be considered F plus, and that gives a bacterium donor status. This says donor status down here, F plus. This cartoon is showing kind of steps of what happens for a donor F plus cell to eventually get a copy of that plasmid into the recipient cell. So if you don't have an F plasmid, you're called F minus. F minus is like just a status that bacterial cells have. And if an F minus cell is in contact with an F plus, then it's considered a recipient. So if you're an F minus cell, you may spend, all your life being F minus and never change. If you're connected with an F plus cell, then you have recipient status until the whole thing is over, and then you too become F positive. Maybe it's like you're not a zombie yet. If the zombie is like biting you, you have possible recipient status, and then if they bite you enough that you become a zombie, then you will be F plus. So there has to be a pilus to connect them. And when this happens, this length of pilus is way too long. But that's just a lot of distance to send DNA through. So when conjugation happens, the pilus joins them, and then it retracts to get them close together, which is cool. Then the F plasmid, which is shown in purple here, will have its DNA nicked, which means there's a cutting process on one of the strands to open it up. So that's DNA nicking. And then there is a single strand transfer. A single strand transfer. It's not both strands that go through the pilus. You only need one. You only need one DNA strand to get the info. One strand moves through the pilus. Then in the recipient cell, second strand is going to be filled in. You complete the F plasmid over here, then the cells go their separate ways. And now this one is F plus as well. This process is part of what we call rolling circle replication. Sometime in my past, I had to sit through a whole lecture on that. Found my notes the other day. Rolling circle replication is a DNA copying method used by bacteria. If you go into, some of you may go into microbiology type PhD work, you can have a whole lecture there on rolling circle replication. I'm not doing that today. But it nicks, it breaks off one strand, it fills in the other strand. That's important in getting this to happen. Conjugation. Conjugation is always going to send a plasmid over to the recipient cell, but sometimes the plasmid does not exist as a circular form. So with this picture, they're showing the host chromosome in green, and they're saying that the F plasmid is integrated. This would contain all the normal F plasmid stuff, but it's joined with the host chromosome. So it doesn't exist as a circle, it's become part of the host chromosome, but it still has the ability to transfer to other cells. And in some cases, the recipient cell can also integrate that chromosome. I feel like I have part of this picture cut off. So what I want to write about this, F plasmids can be integrated into the genomes of host and donor. If they are integrated, they are able to move extra DNA along with them. So see how here's the nick, here's the single strand transfer, and it's more than just the F plasmid that's coming over here. It's extra parts from the donor genome, kind of coming along by accident. So if the F plasmid is integrated, it can bring with it a lot of extra DNA. It's not length restricted if it's coming out of an integrated chromosome. So let's go to our comparison stuff here. Good. And we'll write some comparison about conjugation. Really, it should be on the same page, but I need more room here. So let's write characteristics of conjugation. Conjugation is going to require F plasmid which may be integrated into into donor chromosome. Doesn't have to, but it might be integrated to move from an F plus donor to a F minus recipient and it has to go through a pilus. There's a lot of requirements for conjugation to happen. It's very finicky. Must have F plasmid. Must go through a pilus. This also requires that the donor and recipient be closely related strains. It can't be two random bacterial cells that have nothing to do with each other. They basically are going to be the same type of strain. Got to be related. This can move some really significant lengths of DNA. If the F plasmid was chromosome integrated. Could move some pretty significant lengths of DNA. They've got to be related. F plasmid has to be part of the story, and pilus has to be where it's moving through. All of these methods require integration into the donor. All of these, would require integration into the recipient. Oh, not donor. Into recipient cell for the DNA to be stable. If the DNA moves, it could be like the most awesome piece in the world. If you don't get it integrated, it will get destroyed and not be useful. Must be integrated. So there are a lot of reasons why we care about these. I want to highlight one that is very relevant It could turn off at any time. Hey, Sydney, something happened to the computer.
Okay, yeah, I couldn't figure out what was going on.
It just kind of stopped, like it did on Tuesday.
Okay.
So, there's one board worth of notes about antibiotic resistance, but didn't miss too much.
Okay.
Looks like it already started recording again too. Okay, so what I want to do next is kind of go through some big topics for the exam. Exam will be on Tuesday. Stop! I'm trying to just get a blank page up so I could. Okay. Here we go. Here's a blank page. I will be in here at about 7:30 if you want to start early. We'll be on Tuesday. And the first kind of big topic that will be on this exam is cardinal temperatures So the idea of cardinal temperatures out of the environmental effects, like what's the minimum temperature, optimum temperature, maximum temperature mean? But the bigger thing out of that is oxygen requirements. So for oxygen requirements, the way that is going to be formatted is the tubes, that picture of oxygen tubes is going to be on the screen, like labeled the way it is, A, B, C, D, E. I think there were 5. And then there will be short answer questions in the style of choose all that apply. So there'll be a question, and then you write down, for example, if the question is, which ones of these have oxygen detoxifying enzymes? You would choose every tube that showed that characteristic and write down the letter associated with it. That one, there's going to be multiple answers. So if there's 5 questions, they're not one answer per question. It's a choose all that apply. That would be one of the questions, probably on the first page of the exam. Okay, next big topic is what's the difference between chromosome versus plasmid, and the major differences between eukaryotic and prokaryotic genetics. Being haploid versus diploid is huge in that. The next big question is going to be lac operon function. And you will have the blank cartoon from that lecture in the test to look at as you answer those. And those questions are going to be in the style of, if you have this change in this particular protein or situation, what is the effect on lac gene transcription? are those genes going to be turned on more or less or totally shut off? And that's a big number of points on this test. You'll need to know what are the 5 viral components and life cycle stages. There's also 5 life cycle stages. So know what are the components and which are essential. Like every virus has to have genome and capsid at least. The envelope, the spikes, and the enzymes, some viruses have, some don't. The 5 life cycle stages in order, those are usually, I like ask you to list one of those. Like list blah, blah, blah. We did not say anything about bacteriophage T4. Did have a day about influenza. So the big thing I usually highlight with influenza is drift versus shift. Specifically in influenza and how that affects human health. Genetic drift versus genetic shift. Like, how does that relate to viruses changing over time or jumping species? That will be the big topic about influenza. Coronavirus, this is new for me to write a test about. So if you, especially on this section, you come across a question, you're like, I don't know what she means. I may not know what I mean. Like, come up and ask me about that. It's going to be questions I've never written and tried out before. So, know what are the major components. So, what's its genome style, the S spike, and the N spike. Those are the major components. What are the major systems affected? So like organ systems. How are these variants cropping up and why does that matter? Oh. And I'm probably going to be connecting drift and shift into coronavirus as well. Like with coronavirus, when was shift happening? When was drift happening? How's that relate to our daily life? On vaccines, I would like you to know the basic facts about Johnson & Johnson, Pfizer, and Moderna. Like how are they constructed and what are the differences in those? Since those, you're probably going to get one of those 3 sometime in the next few months. And then genetic transfer. Kind of compare, contrast the methods that they use. And I think I have that one as questions that are like, choose all that apply. A similar kind of thing, like which ones of these involve plasmids? Which ones require integration. Those are going to be mostly short answered. And then I usually have a more thinking kind of question about transformation, specifically with Griffith's experiment. There'll be kind of a bigger, more thought-provoking question about Griffith's experiment. If you haven't, you can still meet with me about your last test. Like if you didn't do as well as you wanted on the first test, and you want to kind of talk about that before this next one comes along, that might be useful. A lot of you have come in to meet with me though, so. That's what I'm expecting it to be like. Any of those things that you have questions about or not sure what I mean by what I wrote up there? Yeah.
Can you go back to the first page?
Mm-hmm. Jamie, can you turn the light half off? Yeah. You just kind of wanted to look at it. Yeah. So these are the big important things out of this section, and some of these will be multiple choice stuff too. But as I look across what we talked about, this is the important stuff. So any questions you have, or there's stuff that you want to review together, I have a good amount of time tomorrow and also Monday that I'm not in class, so.
The last genetic transfer, the transposition, that's not going to be on?
Yeah, I'm not going to have anything about transposition.
Okay.
I just don't have time for that, and I think that gets covered in molecular bio pretty heavily, I think, and I know a lot of people will have that eventually, so. No transposition. Yeah, I think you want to wait a little bit to take it. Okay. Yeah, I was thinking about that last minute. She needs to probably wait a little bit.
So what's the best? So I'm taking an organic test on Monday, and then Thursday I'm taking a people test.
I would think like the next week.
Okay. Okay.
Like the next Monday,
Okay.
if that works for you.
That works. Yeah, that works way better.
Yeah.
When can is there a link online now to set up like a meeting with me?
Yeah.
Okay.
Well, I know I do in one class. I may not have it this class. Let me save this stuff before I.
Okay.
Okay, I've got that one. That one's saved.
Did you any talk that?
Okay, that one's saved. Big transfer. Ooh, may not have saved that genetic transfer. And I probably need to stop recording this. There's so much stuff.
Dr. Long, I
Stop.
won't be here on Tuesday.
Okay,
We don't have class Wednesday,
that's true.
so should I plan to take the exam Thursday?
What about Monday?
I will be here Monday.
You won't be here? Yeah, that's right. You all usually leave on Monday. Yeah, I am not coming here